You're listening to the Interactome, a podcast by a group of young researchers who want to connect you to the world of science by sharing their stories and perspectives. Just in case their bosses are listening, they want to remind you that the opinions expressed here are their own. They also want to remind you not to take anything they say as medical or professional advice, as they are not doctors. Not yet, anyway. Stay tuned about that. And, without further ado, welcome to the Interactome.
Hi, Future Sarah here, and welcome to Part 2 of Episode 2 of the Interactome. In Part 1, we talked about the biology of cancer cells, what they are, how they work, and where they come from. We recommend listening to that episode before this one if you haven't already. In Part 2, we talk more about what it's like to do cancer research in a lab and how treatments are developed. Without further ado, take it away, Past Sam.
So, we've been talking a lot about, how cancer works, but you were saying, we don't necessarily know all of these answers, but I think one thing that we can talk about, or you all can talk about, is what it's like to actually do this research. So what's it like to actually try to treat cancer, or to make drugs to treat cancer, because none of us are doctors.
Yeah, no, I think that's actually something really interesting to talk about, because I think when I was first interested in research, I was just like, how are you able to kind of, I don't know, it's a interesting concept to think, how can you get from being in a lab where everything is sterile and you're at a bench and you imagine people in white lab coats and wearing goggles to getting a treatment that you give in a clinic to patients. And so in my experience, so I'm currently working on targeting a certain protein and knocking down the expression of that protein and seeing what happens to cancer cells. So what we do is essentially we have cancer cells that we've taken from patients, and we grow those in dishes. We literally have an incubator, and thank you to scientists many years back for realizing, what are optimal conditions. And basically we grow human derivatives of human cancer cells in dishes, and then change the expression levels of proteins, or and Joe does different things to cancer cells, and then we measure the changes, so we see if the cells stop growing or if they grow at a more rapid rate. I also work with mouse models, so mice are used often in cancer research. So it's kind of like you work in cells, and if it works, then you move to mouse models. And anything in mouse models is kind of like the step before. Work in mice is kind of the step before clinical trials. So essentially I do a lot of cell culture, a lot of work with mice, and yeah. What about you, Joe? Do you want to talk a little bit about that?
Yeah, I mean, I think whatever I guess for me, maybe it'd be helpful to share with everyone a bit about just what my day is like day by day, just in the lab. I get in there at 9, sometimes 10, really depends. I get in, I check my cells, so I go in and see how my cancer cells are doing. I have cells in dishes for my experiments too. I keep stocks, where I'll actually I'll have extra cells for all my experiments just growing. I'll just see how they're doing, see if I need to replenish their media, which you could tell if the media is more yellow, it means that you need to replace it, things like that. And then I'll go to my lab bench to start my basic non. Oh, maybe I should explain what media is. Media is it's basically broth, almost that literally derived from um, bones and things like that. That is basically, it literally is just the food for your cancer cells. Our cancer cells, they like to They like to live in basically juice almost. Um, and if they don't get that, they're very unhappy. It's very interesting how a lot of the cell lines that we've derived from humans are very, very finicky without their this media. But in humans, they're very, very tough. So that's what.
Yeah. So I always make the joke that like why are my cancer cells being so difficult when they're in a dish? Like they're such brats. They're like, if you don't give me this specific type of media with these specific growth factors, then I'm gonna die. But when they're in humans, they're like, they've taken their pre-workout and they're ready to go.
Yeah. Um, yeah. I think there's still a lot that remains to be understood about cancer, and also there's a lot of questions about the right model or like basic of cancer is, like for our experiments. So for example, like things like cells in a petri dish versus organoids, which are basically like just like cancer cells, but just more in like a 3D shape rather than like a flat surface on a dish or things like that.
So, from my perspective, as someone who doesn't work with anything more complicated than a bacterial cell, wouldn't an organoid be better? Like just inherently, like wouldn't that be like the better model no matter what you're doing?
Yeah, it's just harder to take care of. So it's a lot harder to work with. It's a lot more intensive, and so it's a question of to like what do you need to use to answer your question. So for example, if you're only looking at like how a specific protein mediates the effect of your drug on cancer cell death, which is my case, all I really need is just like cells that express that protein, and that's it. It doesn't matter whether they're in like on a flat surface, or in a 3D shape. But for other cases where if you want to, for example, model like tumor vasculature, or model those like blood vessels, or things like that, in addition to just cancer cells, like maybe having a 3D culture is really good. But a big disadvantage of really both of these models, since they're not actually like humans, is that they're not humans. We don't know exactly if like the like when we see the organoid die, or if we see like the cancer cells on the flat dish die, whether that means that if we treat with the same drug that we've treated these two models with, cancer cells in humans will also die.
Yeah, as a listener, I'm like, okay, so why do we trust you then? Like, why? Yeah.
Yeah, I mean, so I guess isn't that part of like the drug development process? And I just remember seeing a comic at one point being like, when someone says, such and such a thing will kill cancer cells in a petri dish, and the comment's just like, so will a handgun.
I mean, it's true. Like you
like.
Oh, go ahead, Sam. Sorry.
No, I mean, I was just gonna say yeah, we continue essentially. Like, how can we tell the difference between it will kill anything, or it will kill a cancer cell and not something else?
Yeah, the biggest, like, you can really, there are a lot of things that can be demonstrated to kill cancer cells or other things like in not humans. But if you want to kill cancer cells only effectively in humans, the only way to really know for sure is to test it in clinical trials. And there's a reason that so many like cancer discoveries or things like that, like in research papers and things like that, don't end up becoming like a cancer treatment or things like that. It's really, really hard to, like, if you have a drug that works in your cell culture, it's really hard to oftentimes get that to translate to full humans.
Yeah, and I mean, I think that's why there is such. I know in our previous episode we talked about the peer review process, and I think it was Sam who said, the cool thing about science is you can say I did this one thing, and then everyone else is like, okay, well, prove it to me, and show me specifically why that works or why it doesn't work. And I think that is why in cancer research we have multiple models before even thinking about going to clinical trials. We test, for example, in my case for my research project, we're testing what happens to cancer cells when we knock down this certain protein. And then we're testing it in patient-derived cells, so cells that we have cells that have been growing in culture in the cell juice under very specific conditions for a long time. And then we have more recently, patient-derived cells. So these have just been taken out of patients. We put them in a dish, and then we treat them with, then we knock down our protein, for example. And then we also, once we've validated that and done a lot of work at that level, then we take it to mouse models. So I think that's a key step before going to clinical trials, is that we test these specific targets in mice, because mice and humans are not exact replicas, but it's a very one of the best models we have.
Yeah, I think I should just apologize to our listeners, because I think I kind of came off as like a few minutes ago as saying like, yeah, the only thing that works is clinical trials. I mean, like, yeah, to a degree, like you only know that something works with something if you definitely test it there, but you can get closer to knowing whether something actually works or not. So like doing all these things that Lauren just mentioned, like those are absolutely critical. And I just want to apologize if I came off as saying that that's not true.
And just kind of going off of something Lauren said there, it's like you're saying, oh, these cells grow in like in media for a while, but like that can happen for quite a long time, can't it?
Yes, it can, actually. And I wanted to mention this earlier, but I think briefly, and we can go more into ethics in the next episode or in later episodes, but one really important woman by the name of Henrietta Lacks has her cells almost all over the world, probably at this point all over the world, helping out in labs. And there's a lot of extensive history behind that, and unethical treatment, and a bunch of racism, and really not so great things. And I think that that's also important to just acknowledge and keep in mind as we move forward into researching more cancer treatments, especially these more specific immunotherapies, and kind of remember the past and then look towards the future and make sure we don't make those same mistakes. Personally, at this point, I've never done cancer research blatantly, so I've never worked with human patient cancer cell lines. So I'm actually curious for Lauren and Joe, what is the process like these days of getting that consent from the patients and making sure that those cells and those cancer lines are okay to use?
I think it really, usually what happens, like the most important thing, and I'm not a doctor here, and I will not claim to be one, but I can share information based on what I know. The most important thing is that before a tumor is taken for any testing, patients must give informed consent, meaning that they know that their tumor is actually, like, normally how you'll get it is you'll go in for surgery a lot of the times, and you'll have the tumor removed surgically, and it'll be put in like a little tube or something, and then frozen in liquid nitrogen, which is really, really cold. And then given to the scientists who have already previously agreed to work on understanding this tumor. And so, yeah, I think the biggest thing is that patients whose tumors are being taken for testing are they're they're supposed to be giving their informed consent, meaning that they understand what it's going to be used for and why. And there have been cases where that hasn't happened, and that was like particularly in the past. So, for example, with what happened with Henrietta Lacks, her tumor was taken for scientific use without her consent, and so that was previously that is one of the biggest ethical issues with obtaining patient samples. But in the vast majority of cases, like consent is obtained, forms are signed, and if they're not, then legal action should be followed, because it's very important that patients whose tumors are being used for scientific research like consent in the first place to the work. But after that long rant, the tumor is usually then sent over to the lab that has agreed to do that work for the specific test that they've already agreed to do. Yeah, I think one other thing that might be interesting to chat about here is just what causes cancer. Like, I think it's there's it's kind of it's that's always a hot topic, um, and I think it's going to be hard to cover it comprehensively just because we're still trying to understand it. But I think we can get into it a bit.
I think honestly, just the most interesting way to start with this, and something that like I think is something you can do something about, is to talk about where Henrietta Lacks got cancer from. So there's a great, well, I don't know, great. There's a book out there that a lot of biologists read called The Immortal Life of Henrietta Lacks, and it details a lot of this. And she, her cancer cells was, I believe, cervical cancer, and
Yeah.
the most common cause of cervical cancer up until recently was HPV. So human
Papillomavirus.
papillomavirus, which is a sexually transmitted disease that so viruses can do all sorts of different things. If you've seen our "What is a virus" video from like last year, HPV has a nasty habit of sticking genetic information directly into a patient's genome, and then that can just cause cancer because it just wreaks havoc on DNA. If I'm understanding that correctly, I'm neither a virologist nor a cancer biologist, but
Yeah, I mean, sticking pieces of DNA from a virus genome into a human genome where you shouldn't be sticking in random pieces of DNA can definitely mess up the process.
And just to stress, because people talk about this for other reasons right now that are not scientifically accurate, that's very rare. It's very, very rare to have something sticking stuff randomly into your genome. Most things do not do that.
Yeah.
But certain viruses will do that.
Yes. Yeah, I think the as of now, and feel free to jump in here, Lauren in particular. It seems that the major cause of why cells just start to grow rapidly and evolve into cancer, when they shouldn't be doing that, is the accumulation of mutations in genes that help rein in growth, or in accumulation of mutations in genes that stimulate growth. But oftentimes why those mutations occur really varies. And there are a lot of different factors that can contribute to that. So for example, there's like people after like at Ground Zero at the World Trade Center, a large portion of them, like, so for example, people who were in the building when it collapsed, first responders, things like that, because of all the carcinogens or cancer-causing chemicals that they were exposed to, a lot of them had a higher, or this group of people had a higher prevalence of cancer years after relative to the normal population.
And
go
yeah,
ahead, Sam.
Well, to talk about prevalence, I mean, something that I think strikes me as a biologist, and like kind of just listen to how people talk about things, like everything in biology is a numbers game. It's odds, it's rolling the dice essentially. And cancer is the odds of these genes breaking. I mean, that's how you get cancers. Like, so what increases the odds of that? And so, like, to be a little bit ridiculous, I mean, you could, smoke 10 cigarettes a day while working at the asbestos and benzene factory and never get cancer. It would be unlikely because all of those things are carcinogens, but your odds of getting cancer go up, but it doesn't guarantee that you'll get it.
Yeah.
So.
Yeah, I think kind of piggybacking off of that, and, I forget if it was said already, but mutations in general are random, and it is just a numbers game. So like these things, carcinogens, mutagens, anything in the environment, really. Even sunlight, right? We talk about sunlight, that's why you got to wear your sunscreen. Like, even that puts you just at an increased risk of having some form of mutation one day that just happens to turn into a cancer. And I think, largely for this talk, we've been talking about metastatic cancer or malignant forms, but there are also benign forms of cancer, which, from my knowledge, just mean cancer that is still localized to the site where it originally developed or where that first cell originally mutated. So, in terms of prognosis and outcome of people who have cancer, that can also vary.
I had a question about that. Is it okay if I ask it?
Oh yeah, go right ahead. Go right ahead.
Is it known what makes a tumor benign or metastatic?
From my knowledge, I believe if it has left the original site, it is deemed metastatic. Lauren, I don't know if you know the official definition, but I believe that's what it is.
You got it. Yeah. It's if they acquire mutations that
I was more wondering
are you asking like how like benign tumors become metastatic?
Or I'm kind of wondering like what exactly about like if you have a cancer cell, how does it kind of decide whether or not it's going to be benign or metastatic? I can't say it. Metastatic. Metastatic.
Yeah.
Thank you.
I got you.
Joe, do you want to talk about this?
To be honest, I don't know too much about like the molecular mechanisms that make or help a cancer cell decide whether it's gonna spread to other parts of the body or not. Like I that's just not my area of work in cancer research. But there are for sure certain molecular changes that do happen. So they're like cancer cells will change their basically like the way that or the amount like the proteins that are on the outside of themselves so that they can like get through the blood vessels more easily and things like that, or like more effectively like crawl around and just hang out in blood to spread to other parts of the body.
Yeah, there's a process called EMT, which is without going into too much detail, it's basically how cancer cells can suppress certain features that allow them to become mobile and to move. And so it's the idea of, I think cancer cells essentially are always trying to acquire genetic changes that allow them to become more aggressive, to grow more rapidly, and to divide. And so in some cases, our body is able to, shut this down. And in other cases, they evade the checks and balances. And so I think where the cancer cells are able to change over from being benign to malignant or become metastatic is really also, in a sense, a numbers game. In that, they're trying to accumulate as many genetic changes that will allow them to become metastatic. And in most cases, they can. And it's also, it takes time. So originally, you may be diagnosed with breast cancer, but with time, there'll be one of these breast cancer cells that is able to kind of break through, that will acquire genetic changes that allow them to move through the blood to different parts in the body.
Yeah, and I'm going to jump in here too. And I feel like the more cancer cells mutate, the more wacky and different they become from normal cells just in the body. And I think this is also why immunotherapy is such an intriguing topic, is because we can use the immune system, right? Theoretically, we can use that immune system to detect foreign particles or signals of any kind that these wonky cancer lines are releasing. And so if we're able to do that within our body, we can actually use our own body's immune system to fight said cancer, even if it's metastatic. And so one therapy that's out there that kind of relates to this is cancer vaccines. So antigen versus antibody really quickly. So an antigen is any foreign particle that the body says, hey, this is foreign. An antibody is actually a protein that you and I would create in response to a whole bunch of immune system pathways and regulations that initially detect said antigen. So we can use those and a lot of different pathways there, and we can go more into immunotherapy another time too, because that would take a while. But at its core, we can use the immune system to really detect and hopefully fight in the future these really nasty cancer lines.
That sounds really cool.
Oh yeah, I think there's definitely, I think we've come very far in terms of like what we know about cancer and how to treat it and things like that. We've definitely, like, for example, breast cancer is now, if you get it, you're significantly more likely to survive than you once were. But there's still a long way to go. Like, for example, there are certain kinds of cancers like glioblastoma, which is a specific kind of brain cancer. It's the most common kind, to my knowledge. It really doesn't have any good treatments for it besides very toxic chemotherapies. We need a good targeted therapy that could selectively kill these brain cancer cells while leaving normal brain cells and other kinds of cells intact. And we just don't have that. So there's definitely a lot that needs to be done, but I think I'm just very, very excited about what the future holds in this field.
Yeah, I think that there's a lot of exciting stuff, and hopefully we'll get better at understanding cancer, because it's this big, complicated thing, and I think that that's maybe, if you could take away one thing from this episode, it's that cancer it's a really complicated disease, and we here had questions, like those were legit questions we were asking each other. Like there's stuff like I hear about, and then that's about as far as it goes, because it's a very complicated system, and we'll be I guess Lauren and Sarah can talk more about this, but like we're going to be posting more information about this for you all to look at, because I think it's to better understand these sorts of complicated diseases. It's good to kind of get a idea for how to read about and how to better understand these things as they change, and they're changing rapidly. I mean, there are treatments. I know people who have gotten treatments that 10 years ago would have been absurd. So, or not absurd, but like they were just drawn on a whiteboard somewhere at like Harvard or something. So, yeah, I mean, there's a lot more to know, but hopefully you learned a bit from this. I mean, I certainly learned a bit just listening to everyone else here. Like I said, I mean, I'm not really a cancer researcher.
Yeah, I mean, I just feel like the field of cancer biology is really making significant and really exciting strides. And so hopefully, we obviously can't cover this all in a podcast episode, but hopefully if you read one of our digests, we were going to dive a little bit deeper into more specific cancer therapies, so specifically immunotherapies we're going to look at. You'll be able to learn a little bit more.
And I think that, no matter who you are, cancer is something that affects you, whether it affects you personally, a family member, someone you know. At any point in your life, you will come across at least one person who has had cancer. And as horrible as the disease is, I think all the treatments out there, and even just ideas for future treatments out there, there is a lot to still have hope for.
Thanks for listening. We are now hosted on most major podcast platforms, including Audible, Google Podcasts, and Spotify, so make sure to subscribe. We should also be on Apple Podcasts soon. For more content, you can follow us on YouTube at Interactome Media, read our blog post about cancer at interactomedia.org, and join the conversation on Facebook and Twitter at The Interactome, and on Instagram at Interactome_media. We'd love to hear your thoughts about this episode and science in general, as well as what you'd like to see next. Stay tuned for more episodes coming soon. I'm really excited for what we have planned.